Uh, Assalamu alaikum. محاضرة اليوم is the continuation of the components of the humoral immunity, and after we discussed the antibodies, now we're going to talk about the complement system. So we mentioned before حكينا إنه the complement system is part of the innate system. And it is the innate component of our humoral defenses. It consists of approximately about 20 proteins. They are present in the serum, and as is the case with acute phase reactants, these proteins are also produced by the liver. So they are constantly being produced by the liver. In the state of need, you make more, and the concentration may increase a little bit, but you have this continuous production of these proteins. So what does complement means? It means it's complementary. يعني it augments, it strengthens, this is the best way is a complement the other immune system, which is the humoral, our innate or adaptive immune system. So the effects of other components of the immune system, they are especially for antibodies. So complement is an important component of our innate defenses. There are three main effects of this complement system. It has a direct cytotoxic effect on non-self cells. So allografts, مثلا, if you put another tissue into the body, complement system will be activated against these, and they will cause lysis. Bacteria, tumor cells that have lost certain capacities will also be targeted by the complement system. So they can do direct destruction of cells. This is through part of the complement system called the membrane attack complex, the MAC. And this is formed from C5b 6, 7, 8, and 9. So this is the first one. The other is chemotaxis. So this helps in chemotaxis by generating of mediators that participate in inflammation and the chemotaxis and attraction of neutrophils. This is mainly done by part of the complement called C5a. And they help phagocytosis. We mentioned C3b many times before, and we mentioned this before. C3b, and we talked how it is an opsonizer, so we can opsonize and coat foreign objects to help our phagocytes engulf them and eat them. So they opsonize them, they prepare them to be eaten, much like how antibodies do. So these are the main three components. We will add a few components as well. How does complement get activated? How do we activate complement? So activation of the complement system can either be done by two pathways, either by an immunologic pathway. This is called the antigen-antibody complexes. So if you have an antigen-antibody complexes, an antibody has been produced to an antigen, there is an immune reaction. This can activate complement. So that's one way. And then you have non-immunologic pathways. So these contain two pathways inside the non-immunologic molecules. مثلا أشياء اللي that are part of the foreign objects. زي أشياء, things that are pattern recognition molecules. Remember about things that are not ourselves. So they're non-immunologic molecules, but we can determine them as not our own molecules. So this is non-immunologic, and these are the two pathways that we can make them. So out of these two ways, we have three main pathways that have a sequential activation of complement. إيش يعني sequential? So we start from one molecule to the next, 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 and then each molecule, as we are forming them, does a function that adds to the entire system. So the first one is the classic pathway. This is the immunologic pathway. This is the pathway that needs an antibody to initiate it. This is an adaptive pathway. So you need an antibody, you need an adaptive response to activate complement, which is an innate humoral defense. So why is it an innate immune defense? Because there are two other pathways called the lectin pathway and the alternative pathway. Now the lectin pathway, تذكروا ما non-binding lectins. So ما non-binding lectins, which you mentioned that were molecules that are part of the non-immunologic molecules, right? So they're not our own molecules. This is a way of activating complement, and then there's the alternative pathway. We will talk about it in a minute. So these two pathways are our innate immune system, and they do not need antibodies. So that's why there is an adaptive component to this innate system. So if you look at the pathways, now we have three main pathways: the classic, the antigen-antibody complex, right? We have the lectin, microbial surfaces, right? This is present on microbial surfaces. And we have an alternative pathway, مثلا non-specific activators, also present on microbial surfaces, مثلا the endotoxin. So I either produce an antibody, or I have lectin, or I have other surface microbial surface activators. What happens if you can see all three pathways? The first step is to make what is called a C3 convertase. C3 convertase, and this also makes the same thing and then goes to the C3 convertase. So there will be binding from each pathway with different molecules that all of them will end with what is called a C3 convertase. هلا الكلاسيك pathway starts with the first complement protein C1, and then with the help of C4 and C2. So C4, every molecule, مثلا C4 will be broken down to C4A, C4B. C1 will be broken down to C1B, C1A, C1 نفس الشيء. كل شيء will be broken down in the entire complement. من هون وطالع, any complement protein, once it is activated, is broken down into مثلا C4, C4A, C4B, C2, C2A, C2B, وهكذا. So in the classic pathway, we have broken down C4, we have broken down C2 by the help of C1. So now we have C4A, C4B, we have C2A, C2B. The B's, C4B and 4B and 2B, both of those are what is called C3 convertase. And then you have the leftover C2A and C4A. And this happens in both pathways, but the initiator horn is C1. The initiator horn is not C1, is non-binding lectins, which with the protease cleaves C4 and C2 and then end up with the same function as C1. So you end up again with C4B, C2B, which bind together and make a C3 convertase. Now C3 convertase, we will get to it in a minute. Now in the alternative pathway, you have the microbial surfaces, non-specific activators, مثلا endotoxin. They will bind to what is called C3 with water plus factor B with a protease factor D. Both of them will break down C3 into C3A, C3B, and then break down B into BA and BB. So the BB with the C3B, so you will have another form of the C3 convertase. So now you end up with two forms of C3 convertase. This is where all things converge. I want to activate C3. So C3 is an important thing that I want to start the complement activation. So once I have C3, again C3B, BB with now C3 convertase. Now I activated C3, I will have C3A, C3B. So now this C3 convertase plus the C3B that has been now cleaved and activated, I will end up with C3B, BB with C3B من هنا مع الإضافة من هون C3A. I will end up with C5 convertase. And now this happens in the similar fashion for the lectin and the classic pathways. I end up with C4B, 2B, and then when I activate C3 this is a C3 convertase, I will have C3A, C3B. So now we have C4B, 2B, and then 3B. This is a C5, another C5 convertase plus C3A. So now either pathway will activate C5. شو راح يصير ب C5 حكينا كل شيء بنعمله شو بنعمله A و B. So I end up with C5B and C5A. C5A خلينا ننتبه له هنا. This is the chemotaxis factor. This is a chemotactic factor. كل ال A's actually are a chemotaxis factor. هذا وهذا وخاصة هذا. بس أكثر واحد فيهم C5A. رقم 2 C3A. وهذول أقل. So every A اللي هي نحكي كانت left over. These are for chemotaxis. These are chemotaxis factors. They're not leftovers. They're not just nothing. So now I have cleaved C5 into B and A. C5A, this is the chemotaxis. C5B will undergo the formation of the membrane attack complex, which is formed from C5B with 6, 7 بدخلوا مع بعض. بعدين بدخل معهم مع 8 و 9. من 6 وطالع ما في cleavage. ما في A و B. They just all get recruited. So I have C5B and then I recruit 6, 7, 8, 9. C5B with 6, 7, 8, and 9. This is called a membrane attack complex. They form a protein that looks like a cylinder. It will bind to the cell surface with a microbe, and then this will attack its membrane and cause a destabilization of the membrane. وبنعرف شو رح يصير lysis and cytotoxicity وهكذا. That is what the complement system is. This is a reduced form of the figure. The same figure is just a reduced form. We have the classic pathway, the Mannan binding pathway. So Mannan binding lectin with C4 and 2, C4 and C2. This is the C1 with 4 and 2. راح ننتهي with 4B 2B 4B 2B مع بعض. هذول C3 convertase, and then I'll end up with C4A and C2A leftovers on the side. هذا كان C3 with B, so C3B, CBB. It is another C3 convertase. I convert C3 into C3A and B. So I end up with a different form of C3 convertase من هون ومن هون. Leftover C4A and 2A. ليش ال A's هون are not كنا نحكي عنهم شيء كتير مهمين as a chemotaxis and anaphylaxis, because I don't form them in the alternative pathway. So they are mainly from C3E. So C3A here. So breaks down and then end up with C3B. C3B, as we mentioned with other forms, will end up with the C5 convertase, which breaks down C5B and A. A had a cell anaphylatoxin, chemotaxis. C5B, 6, 7, 8, 9 will make a membrane attack complex. All three pathways lead to the production shoe C3B. All of them have C3B. So I will have opsonization from any of the three pathways. I will have C5A, which is an anaphylatoxin from all three pathways. I will have C5B and then 6, 7, 8, 9 from all three pathways. So the presence of C3B on the surface of microbes marks it as a foreign and targeted for destruction. This is mainly basically just the same as IgG and IgM, right? So as they coat cells and with FC fraction تبعهم is what phagocytes bind to. So that's the same here. C3b has two important functions. So the function that we mentioned is the opsonization of bacteria, because the phagocytes have receptors for C3b, much like they have receptors for the FC fragment of the antibody. Now it combines with other complement components to generate, حكينا what? We know it moves C3b, moves the complement pathway through generation of C5 convertase, which leads to the production of the membrane attack complex. How do we regulate this complement system? Now, if it's always active, it will cause damage, obviously, just like any system. So you have to balance it out. Subhanallah, Rabbna, as in everything, is made in balance. So this is also balanced out to limit its activation. So once IgM and IgG are bound to their antigen, they will then have confirmatory change. So they will change a little bit their shape that allows complement binding and initiation. So that means شو? Means complement will not always bind. مش حكينا IgG and IgM can activate complement. They cannot always get activated unless they are bound to their antigen. عشان هيك حكينا antigen antibody complexes are only the way that to activate the classic pathway. So they're not always going to, because IgG and IgM are always going to be present. So if they do not bind their antigen, they're not going to have this conformatory change, which will not activate the classic pathway. So this is one limitation here. The second limitation for the alternative pathway is we have protease molecules that inhibit both classic and alternative pathways and keeps them in check. Now the third one, how do we prevent this complement system from attacking our own cells, our healthy cells? This is important because a cancer cell will not be healthy most likely and will not produce this factor, which is called the decay accelerating factor. It accelerates the decay of the membrane attack complex. فبيخرب membrane attack complex. It will decay and will break apart and will not cause damage. Human cells are protected with this DAF, decay accelerating factor, which is called also CD55. So all of our cells will have CD55 DAF to protect them from complement activation and membrane attack complexes. Now, what are the biologic effects of complement? حكينا first, opsonization of microbes with C3b because of the receptors like we mentioned. Chemotax, as I mentioned, C5a is an important chemotactic factor. C5, 6, and 7, all these attract neutrophils and cause the migration towards C5a. They also help neutrophils attach to endothelium, which, if you remember from your pathology lectures, that the beginning of inflammation, neutrophils will have to attach to the endothelium at the site where they need to start migrating into the site of infection or inflammation. So C5a will help this neutrophil attach at the site of endothelium where they need to start migrating to the site of inflammation. Anaphylaxis toxins, حكيناهم C3a, 4a, and 5a. 5a is the most potent of these. They cause degranulation of mast cells, and you will have the same effect as مثلا anything that activated these mast cells. Just like by IgE or anything else, this is the same factor. You will degranulate all these histamine. You will cause increased vascular permeability, bronchospasm, the same as allergy or type 1 hypersensitivity caused by IgE. So this is very, very important. Anaphylatoxins, C3a, C4a, and C5a. Cytolysis by MAC, we know this. The membrane attack complex will form pores in the membrane, which ultimately causes lysis. We know this. They also enhance antibody production. C3b itself, ذكروهم B binds to B cells, enhance antibody production. All of this will enhance production of antibodies, and this is a loop between the classic pathway. B cells produce IgM and IgG, and then more classic pathway activation. So this will be sort of a ring. Do we have any clinical aspects of complement, inherited or acquired deficiency? If you have deficiency of complement, especially the components from C5 to 8, which is what? The membrane attack complex, you end up with an enhanced susceptibility to Neisseria bacteremia. So complement is very, very important in keeping Neisseria in check. Exactly why just Neisseria is the most important of these. There are many theories وفي حكي بس إنه بالنهاية there's no final answer. We just know that Neisseria becomes very, very important infection in people with an inherited or acquired deficiency of complement. So this is very important. So they are prone to recurrent pyogenic sinus and respiratory tract infection. Why? Because this is in زي ما حكينا, Neisseria is in the upper respiratory tract. This is where the area of colonization for it. Inherited deficiency of C1 esterase. So C1 esterase inhibits C1 basically. So it reduces complement. If you do not have this esterase, so you have an over exaggeration of the classical pathway. So inhibition results in angioedema, which when the amount of inhibitor is reduced, the overproduction of esterase occurs, and then this leads to an increase in anaphylatoxins اللي هم راح يزيد ال C1 esterase, and then you will have the inhibitor, so C1 will increase, and then we have more activation of the pathway, and a lot of C4a. C4a will cause a lot of capillary permeability and edema. If you have an acquired deficiency of the CD55, the DAF, شو راح يصير? Your cells will be prone to the complement mediated hemolysis, يعني destruction. Mainly will happen to RBCs, and this will cause paroxysmal nocturnal hemoglobinuria, which is a hemolytic type of hemolytic anemia. Transfusion mismatch, also allografts. When you put a cell from another body that has antigens that to another person, مثلا transfusion or allograft, this antibodies to the A antigen to the recipient will bind to the antigen of the donor, and then complement is activated. This will release anaphylatoxins, membrane attack complexes, and then the patient will have shock due to the anaphylatoxins, and you have red cell hemolysis due to the membrane attack complexes. Immune complex bind complement, and that's any disease where you have levels are low and are high. مثلا immune complexes مثلا عندك complement levels are low in immune complex disease. شو يعني؟ مثلا types of glomerulonephritis, systemic lupus erythematosus. You have a lot of immune complex mediated. These are both immune complex. Some glomerulonephritis are immune complex mediated, and systemic lupus as well. So if you have a lot of immune antigen antibody immune complexes, you will have a lot of activation of the classic pathway, and you will deplete your complement. كل الكومبلمنت راح يضل مثلا على الجوينت أو على الكدني or wherever the immune complexes are concentrated. This will deplete, راح يخلص الامين الكومبلمنت في السيرم. So this the complement levels will be low. شو راح يصير؟ راح يصير هذا البني آدم راح يكون عنده أولا نقطتين. الأولى the majority of these، لأن صارت تركز الcomplement system تركز البروتينات in that area where all the مثلا systemic lupus مثلا بالjoint, in heart, in lung, whatever you want, those complexes are there. You will deplete all your complement, and then this will attract, as we know, the PNCs, which will all release their granzymes and destroy the tissue around it. And that is how مثلا the kidney will get destroyed in glomerulonephritis and systemic lupus, because the complement will be activated where at the site of the immune complex deposition, basically. Patients with severe liver disease شو راح يصير معهم؟ A patient with severe معهم عنده liver disease, what would happen? They don't have, their liver function is less, so they are producing less proteins. Part of the proteins that are produced less راح يكون شو؟ Complement. So they will have less complement. That means they will be predisposed to infections by pyogenic bacteria, because that's what you need to. This is the C3b will be reduced, which is your optimization will be reduced. And that's it. That's the complement system. I think it was a quick, fun lecture. The next lecture, I think there will be 2 lectures of بالimmuno. I think it is the hypersensitivity, inshallah, and that will conclude the immune system. So we have 2 more lectures. ويعطيكم العافية.